All right, so we're going to get started with section 5.2 notes. This is going to be over meiosis. So we just ended last unit, or I guess last semester, with talking about mitosis and how all of our body cells and everything reproduce. And now we're going to move into meiosis, which is basically like reproduction in like mammals and different type of animals. So quick recap on mitosis. So again, that's the formation of somatic cells. Those are our body cells. And then each daughter cell that is coming out of mitosis is going to have 46 chromosomes. And usually those pair up together, so we call those diploid cells again, or that 2N. And that means that we have 23 pairs of chromosomes in all of our body cells. And so examples of different body cells or somatic cells are like our skin cells, muscle cells, pretty much any cell in our body, hair cells, anything in between. So looking at meiosis now, it's a little bit different. So that occurs in our gametes, and those include our eggs in females and then sperm cells in males. And so these are only used in sexual reproduction. And so each of these gametes contain 23 chromosomes. So they're going to contain half as much as all of our body cells. And so that's what we call them haploid. So you can remember haploid as half. And so it's half the number of chromosomes as somatic cells. And so when we look at that 2N or N, that N just means how many chromosome or the chromosome number. Okay, so review of a diploid cell. So a cell that contains a full set of chromosomes. And so there's one pair of chromosome from each parent in those diploid cells. And then you can see on this top area, haploid only has one half. So again, haploid cell, that's a cell that contains half the number of chromosomes. The gametes are sex cells that have 23 chromosomes, while the body cells are going to have 46. So there's that N and 2N that we talked about. So meiosis begins with the diploid parent cell, and then the end result is 4 haploid daughter cells. Okay, next up we have a zygote. So a zygote is a fertilized egg which develops into an embryo. So basically what we have is we have an egg from a female which has the 23 chromosomes, and then a sperm from a male which has another 23 chromosomes. When those come together, they create a zygote, which that's where we get our 46 chromosome, or all of those 23 pairs. And so 46 is the normal number of chromosomes for a human. And so when we're looking at meiosis, so going back to mitosis, that was just one phase. So we started with interphase, went through our PMAT, which was prophase, metaphase, anaphase, and then telophase, and then cytokinesis, and then it was done. But meiosis, on the other hand, is going to go through basically all of that twice. So there are two divisions of meiosis, so it's meiosis 1 and meiosis 2. So two divisions are necessary because each sex cell contains only 23 chromosomes. So we're going to go over the phases of meiosis 1. So the first half, so interphase 1. So during interphase 1, the cell's chromosomes are replicated or they're copied to create sister chromatids. And then similar to the process that occurs at the beginning of mitosis. Next up, we got prophase 1, so that's when the nuclear membrane breaks down. Spindle fibers begin to form. Sister chromatids form homologous pairs, so that means that the chromosomes are the same size and shape. Those ones pair together, and then homologous pairs form structures called tetrads, which means 4 homologous chromatids or 2 homologous chromosomes. So going back, looking at what a chromatid is, that's when we just have one half. So basically 2 chromatids come together and that makes a chromosome. So a chromosome is a pair of those chromatids. Basically that's all that happening. And then these chromatids, these X's right here, these contain all of the DNA for an organism. So this whole thing contains all the DNA. Next up we got prophase 1. So this is when that crossing over happens. So this occurs when homologous chromosomes, those will switch segments of DNA with each other. And this process is important so that we can increase genetic variability. So this is like a recombination of genes and sex cells, so this is why siblings are genetically unique. Um, so basically you have when the genes from each parent will cross over and you get different genes, and that's kind of how all of our genetic variability comes out of. We got metaphase 1 up next, so this is when the homologous pairs of chromosomes are going to line up together at the middle of the cell, and then the spindle fibers are going to attach to the chromosomes, and we can see now down here that instead of having the exact same DNA, we had that crossing over, so we had different sections of each chromosome kind of switch portions of their DNA. So now we have unique characteristics for each chromosome. And then we got anaphase 1, so this is when the homologous pairs that form each tetrad are going to be separated from each other, and then they move to the opposite ends of the cell, and then sister chromatids are going to stay together during this whole time. And then telophase 1, that's when the spindle fibers will break down, the nuclear membrane is going to reform, and then cytokinesis 1 happens. That's when the cytoplasm splits equally among the daughter cells, and then now we have two cells. So each cell is now genetically different from the parent cell. And then that will start into meiosis 2. So this is similar to the mitosis phases, but it's going to produce 4 genetically unique haploid cells. So interphase 2, during the second interphase, the chromosomes are not going to be replicated, and this occurs simultaneously in both of the daughter cells from meiosis 1. Prophase 2, nuclear membrane is going to break down again, spindle fibers are going to form. Metaphase 2, the sister chromatids line up at the middle of the cell again, or the equator. The spindle fibers will attach again. Anaphase 2, those sister chromatids are going to be separated and move to the opposite ends of the poles, and then the spindle fibers are pulling them in opposite directions. So big thing here is knowing that now that the sister chromatids are going to be separated. Telophase 2, so the spindle fibers are now going to disappear. The nuclear membrane begins to form, and then the cell begins to split apart again. And then cytokinesis 2, so the two cells from meiosis 1 have now divided into 4 new haploid daughter cells. So these still have half of the amount of chromosomes, so each new daughter cell is still going to have 23, and then due to crossing over among the homologous pairs, each of these daughter cells is now genetically unique. And so because each cell has half the number of chromosomes as the original, fertilization occurring between an egg and a sperm cell restores the 2n, or the diploid number. And so we see here, so this sperm cell has its 23 chromosomes and all of its DNA in here, and then when it merges with an egg cell, the purple chromosome and the green chromosome are going to come together in the zygote, which is a fertilized egg now. And then now these pairs are all together. Okay, so where does meiosis occur? So in males, it's in the testes. It's called spermatogenesis. So 4 sperm are produced. And then in females, it's in the ovaries, called oogenesis, and that's when 4 egg cells are produced. And then in summary, so in interphase 1, so DNA replicates and condenses, it'll form sister chromatids and nuclear membrane will disappear. Prophase 1, chromosomes form homologous pairs and crossing over occurs, centrioles move to opposite poles. Metaphase 1, homologous pairs will line up in the middle of a cell along the equator. Anaphase 1, homologous pairs will then separate and chromosomes are pulled to opposite sides by the spindle fibers connected to the centriole. Telophase 1 cell will split, forming 2 new cells. And then in meiosis 2, so again that prophase 2, nuclear membrane is going to break down and the centrioles will move to the opposite sides of each cell. Metaphase 2, sister chromatids are going to line up in the middle along the equator. Anaphase 2, sister chromatids are pulled apart by spindle fibers, and then telophase 2, the cell splits, forming 4 genetically unique daughter cells that contain haploid sets of chromosomes. So again, haploid means half. So here's like a little box to help you guys remember some of this stuff and get it all written down so you guys can know the difference between each type.